Hi biologists. Let's start by looking at the learning objectives for this section. At the end of this section, and following the biology syllabus, you should be able to define growth regulators and describe their transport through the vascular system. Describe combined effect. Be able to define a growth promoter and a growth inhibitor, and be able to give two examples of the use of plant regulators. What does this actually mean? What are we trying to learn? Well, at the end of this section, you should be able to give the definition of growth regulators and be able to describe how they're carried through the vascular system or the xylem and phloem in a plant. You need to be able to describe the combined effect or describe the way two or more growth regulators are used to inhibit or promote plant growth. You need to be able to give the definition of a growth promoter and a growth inhibitor. And lastly, you have to give two examples of the use of growth regulators. Growth regulators. Well, what is the definition of a growth regulator? This is one of the objectives of our lesson, so make sure you learn it well. A growth regulator is a chemical that controls the growth of a plant. In our day, they used to be called hormones. Growth regulators can stimulate the growth of a plant or inhibit growth. If they stimulate growth, they're also known as growth promoters, or they promote cell elongation. Or they can be called growth inhibitors. How are growth regulators transported in the plant? This is another objective of this lesson. Well, regulators are actually transported through the vascular system of the plant. In other words, they travel in the xylem and phloem vascular tissue. Vascular tissue is tissue that's used for transport. In a human, blood would be referred to as vascular tissue because it transports dissolved substances. So xylem and phloem, as we would have already learned in our video on plant anatomy, are vascular tissue, so they transport dissolved substances. Remember that xylem vessels or xylem tissue consists of hollow pipes of narrow bore and are responsible for transporting water and minerals, whereas phloem vessels are responsible for the transport or the translocation of food. As an added extra, if you put on your thinking cap, especially those of you who are able for a bit more, you could actually make the argument that growth regulators are like hormones. This is because they are active in very small amounts. They are produced at a specific site in the plant. They're produced in the meristematic regions or the tippy top of the shoot or the root. Rather like hormones in humans, being produced at a specific site, they are produced by endocrine glands. The growth regulators are transported in a vascular system, rather like hormones in humans are transported by the blood system. In plants, the growth regulators are transported by the xylem and phloem, as we said already. Growth regulators cause an effect on another part of the plant, in the same way that hormones have a target organ or a target site. And lastly, growth regulators are slow acting with a long-lasting effect. Now, what is the definition of growth promoter? Again, this definition is an objective of our lesson and we have to make sure that we know it off by heart. A growth promoter is a chemical that causes increased growth in plants. It causes cell elongation. Examples of growth promoters are auxins. Auxins are a collection of growth promoters. IAA is an example of an auxin. The bad news is, especially if we're able for a bit more, we should remember what these initials stand for or what the acronym means. It refers to indole acetic acid. Gibberellins are another example of growth promoters, as are cytokinins. What are the functions of auxins? Well, auxins cause phototropism and geotropism. You might remember this from our previous video. Tropisms are growth responses to stimuli in the environment. A phototropism is the growth response to light, and geotropism is the growth response to gravity. And auxins are responsible for these responses. Auxins also promote apical dominance, suppressing the growth of side buds. Apical dominance is where the terminal bud, or the bud at the end of a twig, or the end of a shoot, or at the tippy top of a tree, prevent the growth of the side buds. Auxins also stimulate fruit formation. What are growth inhibitors? Just to remind you, the definition of a growth inhibitor is an objective of this lesson. So, a growth inhibitor is a chemical that causes reduction in growth in plants. Ethene and abscisic acid are growth inhibitors. The chemists amongst us might recognize ethene by its older name of ethylene. What are the functions of ethene? Well, ethene causes fruit ripening. It is responsible for fruit colour formation. It's responsible for the way that green tomatoes turn red after a period of time. Ethene causes leaf fall and causes the aging of plants. What are the functions of abscisic acid? Well, abscisic acid causes the production of bud scales. These are modified little leaves or scales that appear on the outside of buds, and they're used to protect the bud. Abscisic acid also maintains dormancy or inhibits germination in seeds. This is where seeds will not actually sprout, although conditions are favourable. Abscisic acid is responsible for this phenomenon in nature. It also plays a role in the closing of stomata, which are the little openings on the undersurface of leaves, and it controls the closing of the stomata in dry conditions. The idea of this is to prevent water loss. Now, what do we mean by combined effect? Well, the combined effect of growth regulators refers to the fact that different combinations of regulators can bring about different effects. Sometimes the regulators work together and support each other. They reinforce what each other's functions are. So this is a synergistic effect. They work in synergy or cooperate with each other. Sometimes the growth regulators can work against each other. This is a more antagonistic effect. Now, these interactions, or the combined effect of growth regulators, can be used by horticulturists, gardeners, and farmers to their advantage. Now, as an added extra, here we have some examples of combined effects of growth regulators. Gibberellins can augment or support IAA action in stem elongation. This is a synergistic effect, as both growth regulators are working in cooperation with each other and encouraging or promoting growth in plants. Ethene causes leaf fall, whereas IAA prevents leaf fall, an antagonistic effect. IAA maintains apical dominance and prevents the sprouting of side buds, whereas cytokinins actually stimulate the growth of the side buds or lateral buds. This again is an antagonistic effect. Now, what are the uses of growth regulators? This is an objective of this lesson. We must realise that growth regulators can be artificially produced outside the plant. NAA, the good news being that you're not really required to remember what the letters stand for. Naphthalasidic acid. Well, NAA, synthetically produced outside the plant, is used in rooting hormone powder. We would have come across this in asexual reproduction when we took cuttings. The cuttings can be dipped in rooting hormone powder to encourage the formation of roots. IAA can be used in tissue culturing. You might recall when we covered tissue culturing or micropropagation that if IAA was added to the nutrient medium, it would promote the formation of a collection of cells called a callus. And ethene is used to ripen fruit. For example, bananas. Bananas are picked green, sprayed with ethene, and are ripened by the time they arrive in the shops. And there you have it. Make sure that you learn your definitions word perfect and know them off by heart. Practice in a jotter. Now that we have reached the end of our lesson, have we achieved our objective? Can you define growth regulators and describe their transport through the vascular system? Can you describe combined effect? Can you define growth promoter and growth inhibitor? Can you give 2 examples of the use of plant regulators?